Hi everyone, in this video we're going to talk about some gastrointestinal diseases. We're going to break it up into a couple of smaller videos to keep it from being too long. And we're going to talk about 3 different types of bacteria: Shigella, Salmonella, and E. coli. First up is shigellosis. So this is the name of the disease caused by the species of bacteria belonging to the genus Shigella. These are gram-negative, non-motile bacilli, so they don't have flagella, they don't move around. And there are 4 different species that can cause disease: Shigella dysenteriae, Shigella flexneri. This one is mostly responsible for diseases in developing countries, Shigella boydii, and Shigella sonii. This one is mostly seen in industrialized countries. So if we have cases of Shigella in the US, it's usually with Shigella sonii if it's contracted here in the United States. The signs and symptoms that people experience with shigellosis are pretty common among all sorts of gastrointestinal distress: fever, abdominal cramping, diarrhea. What separates shigellosis from things like salmonella or other types of food poisoning is the appearance of blood in the stool. And this symptom is called dysentery. If you've heard that word dysentery before, it really just means blood in the stool. It's not tied to any particular bacteria or virus. And this is a great differentiating factor between Shigella and Salmonella. The Shigella species can produce a number of virulence factors, things that are responsible for causing disease. Among them are a type 3 secretion system, which we'll review in the next slide, and a number of enterotoxins. Entero, again, means related to the gastrointestinal tract. So these are toxins that are going to affect the GI tract and cause symptoms there. The most important of these enterotoxins is called Shiga toxin, Shiga like Shigella. In species of Shigella that express this toxin, there's a 20% mortality rate. That means a 20% death rate for people who have this infection. Now, the type 3 secretion system, just to remind you, is also called the injectosome because it directly injects toxins into the host cell. These secretion systems are multi-protein units, and they span the inner membrane of our bacteria and the outer membrane of the bacteria. They are a hollow tube which then inserts itself into the host cell membrane, and molecules like toxins or other types of proteins move through the hollow tube of our secretion system and are directly deposited into the host cell cytoplasm, where they can carry out whatever functions they have. So a number of Shigella toxins are moved this way. Shiga toxin is actually an AB toxin. So it has a B subunit that binds to the cell, and the A subunit that's the effector subunit or the active subunit. And what the A subunit does is stops host cell protein synthesis. It interacts with the ribosomes and inhibits translation. If we cannot carry out protein synthesis, the cells die. It's a very effective, very potent toxin. So let's look at the process of infection. In our first step here, we have that very beginning of an infection process where our Shigella bacteria are going to attach to the epithelial cells of the colon, the cells that line the inside of the colon. But rather than just sticking there, Shigella triggers endocytosis, and our bacteria get taken up into the cytoplasm of the cell. Once in the cytoplasm, they actually begin to replicate. And then a really interesting thing happens. In order to invade more cells, Shigella polymerizes cellular actin. Those cytoskeletal proteins of the host cell are going to polymerize and act like little tails that push the Shigella from the original cell into neighboring cells. This allows the bacteria to spread from cell to cell, but also to avoid immune defenses, to avoid immune response. So a pretty clever way to spread and not get recognized because the bacteria are never leaving the cell, they're just staying within them. As the bacteria continue to replicate within the cell, move from cell to cell, and produce Shiga toxin, the cells will eventually die. And those dead cells create what's called an abscess within the colon. Once these cells die, the bacteria can leave them and get into the bloodstream. But a really good thing here is that pretty much as soon as Shigella enters the bloodstream, it gets phagocytized. So it gets taken up by immune cells and destroyed. This means there's no bacteremia. And bacteremia means bacteria in the bloodstream. And this does not happen with Shigella, which is different from some other bacterial pathogens. Here we have a picture of Shigella, again, a gram-negative rod with no flagella. And Shigella is responsible for about 90 million cases of gastrointestinal distress each year and about 108,000 fatalities annually. Most of the cases in the US are travel associated, which means someone went to another country for a trip, for a vacation, picked up Shigella, and then came back and started experiencing symptoms. We can pretty easily diagnose Shigella. There is a rapid diagnostic called XTAG, Gastrointestinal Pathogen Panel. It's a nucleic acid test, so it looks for the presence of DNA or RNA associated with a wide variety of gastrointestinal pathogens, bacteria and viruses. If a person is diagnosed with Shigella, usually we call it a self-limiting disease, which means it will clear on its own, or that your immune system will control it and get rid of the infection on its own. So no antibiotics are generally. Instead, what we would do for our patient is give them supportive care. This is basically what you would do anytime you're sick with a diarrheal disease. Push fluids, make sure that your patient stays hydrated, and try to get electrolytes into their system as well. In really severe cases of Shigella, we can prescribe antibiotics. Mostly it's this one called ceftriaxone. It works to shorten the duration of disease so our patient isn't sick for as long. And it can also prevent spread between close contacts, so for caretakers and for family members.